So this is the talk about like a summary of the latest paper in the life series that I wrote. And so it will be life paper 13 about synergies between life and hab worlds. All right. Okay, so eventually, if all goes right, we will be able to see something like this. And this will be the Earth that we see here. So we would be able to identify directly imaged planets, but they will really be very unresolved spectrographs. We will be able to then see a lot of features from the spectrum. And these features are different with respect to the wavelength range that we are observing. In the top panel here, you see the reflected portion of the spectrum. So it's light that comes from the star and that's scattered back out from the atmosphere and the surface of the Earth. And then in the bottom two panels, you see actually the emission of the Earth by the fact that the Earth and its atmosphere have a temperature. And this is in the mid-infrared. So in the whole scheme of things, hab worlds would also HWO hab worlds independently. The Habitable Worlds Observatory will focus on the range between 0.2 to 2 micron, more or less. So the reflected portion of the spectrum, while life, as we know, will focus on the mid-infrared range. We're still very unsure on exactly where the stops of where are we gonna focus our attention in both of these missions. We are very much discussing this, but this is the general idea that we are sure that they will span two different and complementary sides of the electromagnetic spectrum. And as Björn mentioned in Life Paper 3, and also circling back to previous papers, we expect to have synergies and complementarities between the two missions. Here in this image, it was still even called Luvar and Habex. We didn't even know it was supposed to be called the Habitable Worlds Observatory, but these were the concepts that came before that then funneled into the Habitable Worlds Observatory. So it's practically the same. And as we can see here, the Life mission would have access to CO2 and CH4 potentially, and then a better estimate of the surface temperature, while habitable worlds would have access to O2 and other molecules that will be detected by both missions in different ways, right? So what we want to do with this paper was to try and understand what was the potential of the two missions separately, and then try to assume that we will observe the same planet with both missions, and then what information could be retrieved from that, having access to both of these wavelength ranges. And then we also study the impact of the noise and how much the uncertainty on the spectrum played a role in characterizing the result, in having better results. Now, for the purpose of this talk, there is no time for me to go through the impact of the noise. Feel free to ask later, but I'll read the paper. There have been some, we made some different models with different noise instances so that we could assess that. So the way we did this, we picked a cloud-free Earth-like planet, very simplified, just to give like a proof of concept. And then we assumed that I have observed this with hab worlds, with the baseline requirements for both hab worlds and life, but we assume to have observed it with hab worlds only, and then life only, and then both together. And you will see here that in the rest of the talk, I will have attached these three colors to the three kinds of models. So magenta will be all the HWO related things, and the blue ones will be all the life related things, and the hab worlds plus life are the yellow gold ones. And the way we do things is through retrievals. I'm sure you heard about this a lot, so I'm not going to spend a lot of time going through this. But essentially, we simulate a spectrum So we start from a theoretical spectrum, we attach some realistic noise instance to it, and we generally use high fidelity noise simulators, so which could be LiveSim, or from the Hub World side, we use PSG, which is somewhat set up on LUWAR, essentially. And then from this simulated observation, we run a retrieval, which tells us what are the subset of parameters that explain the data best. And we are interested especially in surface temperature and pressure, mass, and then pressure temperature profiles to define the atmosphere, and then the composition of the atmosphere. And these are the results. When it comes to the pressure temperature profiles, we see that Hub World here it doesn't do a good job in terms of defining the pressure and the temperature at the surface, but that's sort of to be expected, because as you remember, the wavelength range that the Habitable Worlds Observatory focuses on is the reflected light, and so there is no access really to the structure, the thermal structure of the atmosphere, or not an easy access rather to that. And this is quite the opposite in the case of life. This is kind of where life shines, you know. So life is exactly what's good at measuring. So we can measure the atmospheric profile pretty well. And then if you merge the two together, you get the best in both scenarios. So you have a much stronger constraints around the troposphere here, and you also have smaller uncertainties to some extent. When it comes to the chemical composition, let's just go between HWO and life separately and then see how they behave together. So both of these two missions either have unconstrained or upper limit only for methane, CO, and N2O. This is granted it's at Earth's abundances. We have had plenty of studies in the life series and in the context of HWO, which obviously detect these molecules at higher abundances. And when it comes to the bulk scatterer, so the bulk of the atmosphere, O2, and the Rayleigh scatterer, in the Earth's case, N2, Hub worlds can detect that much easily. And that's obvious in the sense that, well, they have access. The Habitable Worlds Observatory has access to the Rayleigh slope that life does not have access to. So that will be why it's easier for that mission to detect that. When it comes to CO2, life can detect it pretty well. Hub Worlds is struggling a little bit. There is not a lot of strong lines in that range, but we have a lot of lines in the mid-infrared instead, like the 50 micron band. And both missions can detect O3 and H2O. But all of this is noise dependent. As I said, trust me on this because I don't have time to go through it, but you can ask later if you want. But when it comes to Habitable Worlds Observatory and life, you see that anytime a single mission detects that molecule, then the two missions together detect the molecule. And when both missions detect the molecule, you get a detection that's higher of higher quality, if you will. So you can see this also in this plot here, where we use the sigma value to estimate how confident we are on each detection. And you see, especially like in the water or in the ozone, that both together will give us even more confidence on the detection. So this is the take-home message that I have. The two missions separately have access to two different areas of the spectrum, and therefore to very different features. And so joining them together, you only improve the results because you have access to more information and you can constrain your information much better. So that's why you should love both Hub Worlds and life. And that's it from me. If we have time to talk, I have prepared a slide on future work that we could think about as the life science team to keep working on this if you want.
Awesome.